Hi, it's Dr. Phoenix. Today we're going to start our study of DNA and RNA. DNA and RNA are nucleotides, and they're extremely important molecules. DNA primarily resides in the nucleus and carries all of the genetic information that codes for all of our traits and our body parts, and as well as all the proteins that we're made from. And RNA basically carries out the commands of DNA. So let's look at the components and what DNA and RNA are made from. First of all, they are made up of smaller building block units or monomers called nucleotides. A nucleotide is made up of 3 parts. In DNA, the nucleotide is made up of a sugar called deoxyribose. There is a phosphate group and what's known as a nitrogenous base. So a sugar, a phosphate, and a base. Of those bases, there are 4 possible kinds of bases in DNA. Those bases could be either adenine, guanine, cytosine, or thymine, and we tend to just call them by their first initial. So A, G, C, and T. Now these nucleotides can be joined together in any order, and it's the order that these nucleotides are arranged in that actually determine the special code that DNA is carrying. It is sort of like an alphabet that spells out very particular messages that's stored in this particular base sequence. So if we take the DNA spiral and flatten it out and look at it in just 2 dimensions, we're going to see that the DNA molecule kind of looks like a ladder. And the ladder is made up of repeating units of nucleotides. This right here is a nucleotide. That's your sugar, here's your phosphate group, and this T is your base. So these nucleotides can be arranged in any sequence, and the sequence does have meaning. These letters spell out certain messages. But as you can see, the shape of this molecule looks a little bit like a ladder. The sides of the ladder, or the backbone of DNA, is made up of the sugar and the phosphates. The sugar and the phosphates make up the sides of the ladder. The rungs of the ladder, or the steps of the ladder, are actually made up of the bases. Okay. Now a scientist by the name of Erwin Sharjah, some years before Watson and Crick came along, in analyzing DNA, noticed that there was always approximately the same amount of adenine and thymine, and there was always approximately the same amount of guanine and cytosine. His discovery was referred to as Sharjah's Rule. They had no idea why this was the case, but nonetheless, this was his observation. Some years later, along came Watson and Crick, who ultimately discovered the overall shape and composition of DNA. And Watson and Crick realized that there are hydrogen bonds that form between these bases. And these hydrogen bonds can form only between certain base pairs. In other words, adenine can only bond with thymine, and guanine can only bond with cytosine. So A with T, C with G. A with T, C with G are the only combinations. Those are the only pairs that can bond together. And Watson and Crick called this complementary base pairing. All right, once again, A can only bond with T, C can only bond with G, and there are no exceptions to that rule. Well, this basically confirmed what Sharjah found, in that there will always be the same amount of A and T in DNA, and there will always be the same amount of C and G because they have to pair up. So ultimately, Watson and Crick realized that DNA is made up of nucleotides, sugar, phosphate, base, bonded together with the bases, complementary base pairing, A with T, C with G, and then the DNA molecule ultimately took on a twisted ladder shape, a double helix, if you will. All right, now the second player in all of this is RNA. RNA is also a nucleotide, and in some ways RNA is very similar to DNA. However, there are three main differences. RNA is made up of nucleotides. RNA has a sugar, a phosphate, and a base, just like in DNA. However, the sugar is different. In DNA, we saw that the sugar was deoxyribose. In RNA, the sugar is going to be called ribose. Thus, its name, ribonucleic acid, instead of deoxyribonucleic acid. The second difference is that RNA has no T's. There are 4 bases in RNA, just like in DNA. However, there are no T's. Instead of thymine, RNA has what's known as uracil, U. Okay, so when complementary base pairing comes along, when something in RNA needs to match up with an A, since there are no T's, U's will match up with A's. The third difference between DNA and RNA is that RNA is a single strand rather than a double strand. All right, so anytime you see RNA, you're going to see just a single strand. Now, keep in mind that you could be shown just a segment of DNA on a quiz or a test, just a single strand, for instance. That doesn't necessarily mean that I'm showing you an RNA strand. Please look for the bases. If you see a T present in the strand that I'm showing you, then of course that would have to be a piece of DNA. If you happen to see a U in that strand, then it would have to be RNA. All right, so those are the basic components of DNA and RNA. We're going to look at how they function, what they do in subsequent videos. So I hope that helps, and we will look forward to seeing you in class tomorrow. Have a great day.